All here tonight. My name is Dr. Jennifer Glowinka. I am the Senior Vice President for Academic Affairs and Dean of the College here at Carroll. And it is my pleasure to welcome you to the Student Research Festival keynote presentation. So our Student Research Festival, which we call SRF for short, is an annual event that showcases student research in collaboration with faculty mentors, honoring their achievements across the arts, sciences, humanities, and social sciences. I encourage all of you to join us tomorrow in the Campus Center for poster sessions and for oral presentations, where students will share their impressive research that they have conducted in their time at Carroll. And in particular tonight, I want to give a special thank you to the Dr. James J. Mannion Endowed Chair of Biological Sciences, to Dr. Russ and Sharon Bell, and to Touro University for their generous financial support of SRF this year. Tonight, it is my great honor to introduce our SRF keynote speaker, Dr. Bryan Rother, and to present him with one of the most prestigious recognitions our institution offers, the Alumni Academic Achievement Award. This award is a celebration of excellence, perseverance, and a lifelong commitment to learning. It recognizes alumni whose academic contributions have not only advanced their field, but have brought pride and distinction to our Carroll community. Dr. Rother is the Michael Hooker Distinguished Professor of Pharmacology at the University of North Carolina, Chapel Hill School of Medicine. Dr. Rother grew up in Butte, Montana, and received his bachelor's degree in chemistry and biology from Carroll in 1977. He received both an MD and PhD from St. Louis University in 1983, and then trained in pharmacology at the National Institutes of Health and in molecular biology and psychiatry at Stanford. Prior to his positions at UNC, Dr. Rother was a professor of psychiatry and biochemistry at Case Western Reserve University School of Medicine, where his clinical specialty was treatment-resistant schizophrenia. Dr. Rother's contributions to medical science and treatment of disease cannot be overstated. And my brief remarks here will not do his work any kind of justice. But he has been engaged in therapeutic drug discovery for decades and has served as an advisor to most of the major biotechnology and pharmaceutical companies. He has developed many new technologies, holds many patents, and has published more than 500 papers in the general areas of molecular pharmacology, structural biology, and synthetic biology, including in the highly reputable journals such as Science, Nature, and Cell over the past decade. Dr. Rother was elected to the National Academy of Medicine in 2014 and the American Academy of Arts and Sciences in 2019. He's received numerous honors and has given many prestigious lectures, including recently to the 2024 Nobel Forum. Dr. Rother exemplifies what it means to be a scholar, a mentor, and a leader. His achievements remind us that the pursuit of knowledge does not end at graduation, it only begins. He embodies Carroll's motto, non scholae sed vitae, not for school, but for life. Please join me in congratulating Dr. Bryan Rother as this year's recipient of the Carroll College's Academic Achievement Award. Dr. Rother's talk tonight is entitled The Promises and Perils of Psychedelic Pharmacology for Psychiatry. Per Carroll's protocols, I remind you that Carroll College seeks to provide a campus intellectual environment that encourages the pursuit of truth, critical thinking, responsible decision-making, civil debate, and respectful dialogue. Carroll College neither agrees nor disagrees with the views expressed, but rather provides a forum so that each person attending may hear, contemplate, and discuss the ideas and opinions presented. Dr. Rother, thank you so much for being with us tonight, and congratulations again. I know how important connecting with the students is to you, and so they are looking forward to your talk as we are.
Yeah, thank you. The only thing, thanks so much for the kind introduction. somebody could give me a laser pointer, that'd be wonderful. It's, yeah, it doesn't,
Just turn it on. Turn on the side switch.
oh, I gotta turn it on.
Sorry.
Oh, there it is, okay. Ah. It's wonderful coming back here. I haven't been here for a long time. The only thing, you know, I hope to come back sometime again in the future. And be wonderful to spend a day with the students. That's the only thing that I think it's good. All right. Can everybody hear me? Yeah, okay. So this is what I'm going to talk about. Before I start, the university requires me to give my disclosures. The one thing I'd like to point out is we have this drug that's in phase 2 clinical trials. It'd be great if it makes it. And then the thing that that I'm most happy about is I don't know the professors. Have the professors here heard of badging? And the professors here. So if you haven't heard of it, you might want to look into it. So you can basically any plasmid you want, you can purchase for $65 from badging. And they have, you know, sent out 59,000 orders of mine to date. I asked them for the statistics last night. So that's for scientists, that's a big number. And then everything I'm going to present today is supported by your tax dollars. So thank you. I'll start out here with this. So when I was here, which was a long time ago, I sort of knew that I wanted to do what I do now, but I didn't know how to get there. And no one here knew how to get there either. But I was encouraged to go into pre-med and go to medical school. And literally 50 years ago, there was a visiting lecturer from Oregon. And he gave us, to this date, I don't know why he came to Carroll College. I think now, so I talked to people in Oregon who may have known who this person was. And the only thing we come up with was he was an avid fly fisherman. He wanted to write this off as a business. But at any rate, he came and gave a lecture. And his lecture was from this book. You can see the other Dr. Roth here. And it was on this diagram, basically. And he was explaining drug action and neurotransmitter action in the brain. And he got to the key point here. He talked about receptors. And literally, it was like I was struck by lightning. And I said, that's what I need to study, is receptors. And if he would not have come and talked to the students, I would not be here. For better or worse. I mean, that may not be a good thing. So this is my diagram. And at the end of the talk today, and this is talk for the students, but I have sort of made it so other people will understand. But this is what we're going to go over. We're going to go over psychedelic drug action. And hopefully at the end of this, you will understand that. And maybe somebody will be inspired. Like this guy here. So the story is, when I was at Case Western, I was still practicing psychiatry. And this was around 2000, this is like 25 years ago now. And a student came to me who had an experience very similar to what you're going to see here on the video. And this was a drug no one had ever heard of before. And he came to me as in my role as a psychiatrist, because he was afraid he was going to go crazy. Psycho. And so I checked him out and he was fine. And I think I said something like, go no more and go and sin no more, my son. But I said, so what is it? What is it you took? And he told me, and I'll tell you the story here, but let's see the experience first, okay? So this is a video I just downloaded from YouTube.
Yeah, I don't say Ashland, but Saudi kind of.
So for those of you who don't know, this is a bomb. See, I said this was going to be entertaining. Okay, so this is a bomb. This is one hit from the bomb, okay?
Give me a thumbs up if you want to feel it. You can get a feel about that.
This is a college student. It's not a Carroll. It's not a Carroll college student.
Yeah. Oh, shit. Yeah.
That's where I usually stop it. So if you go to the end of the video, he's fine. Okay, the whole experience lasts about 5 minutes, basically. But pretty intense. So I asked the student, I said, so what is this thing that you took? And he said he smoked salvia, which I had never heard of before. And so he ordered it, this is 2000, ordered it on the internet, basically. And it was sort of big in the early 2000s, before marijuana was legal in most states as a marijuana substitute, basically. But most people didn't like it. So I contacted this gentleman here. His name is Daniel Siebert. And he was trained as a chemist at Berkeley. And he is the first person who, in the world, who purified the active ingredient from this plant and took it. Okay. So this is the first human account. So the compound is called salvinorin A. And you can see that he has this really unusual experience that we call spatiotemporal dislocation. So people are transported more or less instantaneously to another space and time, okay? And usually when they're transported there, they have forgotten that they've taken a drug. So it can be extremely terrifying. But for Daniel, he said, Eureka! I have stumbled upon the psychedelic essence of Salvia divinorum. So, so this is the plant itself. It looks like salvia that grows in your garden, but it is not. Okay, it's a different type of salvia. And this is the active ingredient. Any chemists here tell me what this looks like to them? Any little like cholesterol, maybe? Yeah, like cholesterol is technically a diterpene. So we looked, when he was in my office, we looked at structure up on the internet, and I said, there is no way that compound is psychoactive. It doesn't have a single nitrogen in it, right? It cannot be a psychoactive compound. And he said, it is psychoactive. So that was my inspiration for the story I'm going to tell you today. So at the time, I was running, which I still run, a program sponsored by the National Institute of Mental Health called the NIMH screening program. And what we do is we screen drug candidates, for their actions at what we call molecular targets. Okay. And so this is basically drug screening. And not to get into too much of the science here, but the way we do it is unique. In that we have the capacity now to screen hundreds and hundreds of receptors simultaneously. So we developed this technology. And basically every what's called G protein coupled receptor in the body, there are like 800 of them, you can screen them all simultaneously. We have chemical libraries that we screen in a parallel fashion. So typically in drug discovery, you would have a chemical library that you would screen against one receptor at a time. And what we do is we screen hundreds or thousands or hundreds of thousands of compounds against thousands of targets in a parallel fashion. So that's the technology my lab invented. And for those of you, and these are the bona fides, basically. These are all my papers with that, some of the papers with that technology. So, so what do we do? So we have this library of receptors from the brain. And so the idea was we'll just screen this compound against all of these receptors and see which one it hits. And by comparison, we're going to screen LSD, right? And so we did that. And these are the results here. So you can see salvinorin A, there's only one activity here. You can see LSD here, which I'll get to later, hits dozens and dozens of receptors in the brain. It turns out that the effects of LSD are mediated only by one of them. But this compound is very unusual in that it hits only one site. So in the process of obtaining this compound, it was not commercially available. I had to obtain it from Daniel Siebert. So I sent him an email and I said, can you send us 10 milligrams of salvinorin A? And I talked to me on the phone. And he said, sure, I can send you 10 milligrams, no problem. He said, I only have one question. Is this for recreational purposes or research? And I said, it's for research. And he said, in that case, it's free. Okay. So he sent us 10 milligrams of it. We tested and we found this. And then we were getting ready to write up the paper. And I was going to acknowledge him for his gift of salvinorin A. And I told him, it's the kappa opiate receptor. So this is so you've heard of opioids, right? So opioids mediate their effects by receptors in the brain. And this is not a receptor that mediates the action of morphine. It's a different receptor. And it was known to be activated by psychoactive drugs. So it was a surprise, but it wasn't that much of a surprise. And I said to him, well, we know the psychedelic essence of salvia divinorum is the kappa opiate receptor. And he said, it cannot possibly be the kappa opiate receptor because he had this idea that this was like a novel psychoactive compound, must have a novel target. There must be something really amazing about it. And he said he was going to test my hypothesis. Okay. So he sent me an email. Notice the date. 27 March 2002. Okay. So the idea was he could just pre-treat himself with Narcan as an opiate antagonist and then smoke salvia. That was the idea. Oh. And I said, this is like a really bad idea. Please don't do this. I was concerned there could be some drug interaction that could kill him. And then I was also concerned that he would die. And then the coroner would come and look at my email urgently.
Yeah.
He said, you have to go through a human use committee, all that stuff. And you can read what he said. He said, if I was worried about that, I never would have discovered it in the first place. So he's a believer in what we call participant pharmacology. Okay. So I went home that night, came in the morning, boom, next morning, he did the experiment. He said, yeah, it's the kappa opioid receptor. You're right. And I told him we were not going to be able to put those results in the paper. But if you read the paper, it says unpublished observation. So one other thing about this, so this is like, I mean, when you study psychoactive drugs, these are the people who work with them. So, I brought everybody in my lab to my office, and I had them read the email. And I said, this is the only time in your life where you will make it a scientific discovery in the morning, and it will be validated in humans the next day. That's true. So, and this is just confirmatory stuff for the students. So there's no, I mean, there's no question. That's this. So, in the time remaining, I'd like to talk about psychedelics in general. And first, the promise of psychedelics. So, as I'm sure many of you know, psychedelics are showing really dramatic potential for treating very serious mental health and substance use disorders. To give you a sense of the impact of this, I just wandered around Helena today and talked to somebody in a shop, and mentioned that I was going to be giving this talk. And this was a veteran who had severe post-traumatic stress disorder, which was cured. So he said when he left the VA, he was on 6 different medications. None of them did anything. And he just volunteered this for me. Okay. But he said he took psilocybin under carefully controlled circumstances, many tries, not just once. He's cured, basically. And I hear, I'm not recommending this at all, but we hear this a lot, actually. And I was telling Patty, my old friend from college, that the psychedelics must be different now, because I didn't meet any people that had these life-changing experiences. Not psychedelics, but these are the sort of conventional psychedelics, LSD, psilocybin, mescaline, DMT, and they're really showing potential to treat serious mental health disorders and mood and substance use. And this is sort of a classic study showing a single dose of psilocybin basically ameliorates treatment-resistant depression in a cohort of patients. And you know, you can pick up basically it's in the, you know, psychedelics are in the media all the time. Excuse me, this is a nice article from the New York Times about how LSD saved this woman's marriage. Here are the papers. This is actually a really important one. This is a study showing that MDMA, also known as ecstasy, which is not a psychedelic, but is similar, shows real, I would say, dramatic potential for treating post-traumatic stress disorder. Basically 2 doses, and a lot of people are fine. And for those of you who are clinicians, this is the data. So you can see there's this big effect on day 1, which is more or less maintained. So they basically get a single dose, and then, you know, most people are fine after that. But the antidepressant activity, you know, maybe is a little bit better than conventional tricyclics and SRFs, although it's still not clear. There's sort of a hint that there might be a greater degree of efficacy, but certainly the fact that you just have to take 1 or 2 doses is a big plus. So a single dose of psilocybin, there's nearly an immediate antidepressant effect. It usually lasts for several weeks. There have been follow-up studies for a year, where people are basically no longer depressed a year later. It appears to be equivalent or perhaps better than conventional treatment. It's not approved for use. The thing that's, and I was never interested in these for their therapeutic purposes. I was interested in them because they change how we view reality. And I thought that was like a really interesting scientific question. One of the things that's very unusual about psychedelics, particularly if they're taken under, appropriate circumstances, not at a rodeo or something like that, or a heavy metal concert. Probably not a good idea to take LSD. But individuals describe the experience as being one of the best experiences and one of the most meaningful experiences in their life, right up there with marriage and childbirth. There are now positive phase 2 clinical trials, so we're hoping that the phase 3 trials come through and it'll be approved. And I'll just say parenthetically that administrations come or go, but one of the remarkable things about this particular administration is they're really interested in psychedelic therapy. So RFK Jr. is a huge advocate for psychedelics, and of course Elon Musk takes them. So he is a world's richest man, so there's something to it. But I suspect these will be approved in the next 5 years. That's my guess. But what is a psychedelic drug? So there's a very technical definition. So this is the definition that I prefer. And this is the definition preferred by the FDA, the EPA, and elsewhere. So it's a drug with LSD-like effects in humans through a particular receptor, 5-HT2A. And for reasons that are not particularly important here, when I did my postdoc at the NIH, I started studying that receptor for the only reason that nobody knew anything about it. And it just turned out that it was like this would turn out to be this really important receptor. So I got some luck there. But this is sort of the history of psychedelics. So you can go back to the sort of pre-modern era, and there from a historical record, it's clear that that people took psychedelics mainly for spiritual purposes, not recreational purposes. And then, of course, in the 60s, they became widely used. And these are just different important things on the timeline. My contributions are here, basically, in this latter part here. So we discovered all of these things. So I don't, when I say I study psychedelics, I don't like take them. We study them in the test tube. But psychedelics are a class of drugs that are called hallucinogens. So that includes psychedelics like LSD, what is that, psilocybin and mescaline. And then there are dissociative agents, and these are drugs like ketamine. So a lot of you probably heard of ketamine for treatment-resistant depression. Can be extremely effective. In the popular press, it's sometimes called a psychedelic. It is not a psychedelic. It's a dissociative agent. And then you have what are called deliriums. So as a psychiatrist, all of these things are like super important because they tell us about. And these are natural products. And then we have these really interesting compounds that include salvinorin A and ibogaine that are called onerogens. And this means they induce a dreamlike state. Although people who take salvinorin A and ibogaine say they induce a nightmare state. So it's somewhat dreamlike. And this classification is really good for those of you who are clinicians, because we know the molecular targets for each of these, okay? And it's clear that they have distinct pharmacologies. And they also have distinctive effects in humans. So this is a video I downloaded from YouTube. This is, if you go to YouTube, it's entitled 50-year-old housewife takes LSD for the first time from the 1950s. So I took that title off because it's a little, I don't know, we don't talk that way anymore. So I would say 50-year-old homemaker. Stop. So person, 50-year-old person.
Yeah.
We wouldn't say wife or housewife. Anyway, let's see. So this is not Excel. You can see the molecules. Wow. This is not a whopping dose of LSD that she took, but you get the idea. So she had a pretty good experience. And this is ketamine. So this is from a CME presentation that I downloaded. This is 20 minutes into the ketamine treatment. And this guy is completely dissociated.
How's it going? How's it going? What are the thoughts that are in your mind?
That we're all connected, that we're all together, that like we.
All right, so those are, different things, basically. And Salvador and you already saw the data. Okay. So there is some merit to this classification. So most of the people who study psychedelics dump all of these drugs in as psychedelics, but I'm just telling you they're not psychedelics. Okay. So I'm going to discuss a little bit about my work. I'm going to get eventually to it. But first I need to tell you a little bit about the brain. That is a cartoon of somebody's brain glowing in the dark. And the brain functions by means of neurons, which encode information. And the person who first discovered the cells the cells in the brain is called neurons. This guy here, Santiago Ramon Cajal, 1906. And there he is. That's the little microscope that he used to discover neurons with. Okay. And I'm going to show you some of his drawings here. So he invented this way of staining cells in the brain using silver, which made these remarkably detailed, although very sparse images. So it's this is technically what's called a Golgi stain. And it doesn't always work. And you're lucky if you get one neuron in the brain, basically, that looks like this. So you get these beautiful neurons. And I want to point out these little protuberances. These are called spines here. And Ramon Cajal thought information probably flowed from one neuron to another through these spines here. Okay, that's what he thought. It turns out he was right. So now we know through research supported by your tax dollars that there are hundreds or thousands of different types of cells in the brain, literally. And it's very complicated. And you can see one of these is called the Cajal-Retzius cell. So that's a cell that he discovered. And so what we're gonna do is we're gonna try to find which of these cell types is responsible for the action of psychedelics, okay? But before I get to that, I'm gonna show you this remarkable video here. This is not mine, to give you a sense of how complicated the human brain is, okay? So this is something that was just published in Nature, not my lab, by the Brain Initiative. And what they did was they took a very small section of a human brain, 1 millimeter cube, okay? So this is smaller, this is like the tip of a pen, okay, a cubic millimeter. And they sectioned that for electron microscopy and then reconstructed the image. So this took 10 years and like $100 million. Okay. And this is 1 cubic millimeter, okay? And what they found was that in this 1 cubic millimeter, there are 100,000 cells, okay? And this is the reconstruction of it. I found it quite amazing. So this is the whole cubic millimeter here. See, there are individual cells there. And they used artificial intelligence, of course, and machine learning to identify all the different cell types. They found in a single column there, 1,800 cells here in a column in the cortex. They were able to describe them all, expert annotation, and you can basically download this from the net. You can look at it on your own. It's really cool. And now we're going to zoom up. And here you see individual neurons here. And they're able to classify those using, this is using, for those of you who use this is using UMAP. UMAP? Anybody here uses UMAP? Just a way of classification, large data sets, commonly used in AI and ML. And then they can predict the cell type for these 100,000 cells and look at them in great detail. Okay. So this is a cool resource. And you can see there it's populated. So it's really, really amazing. So I'll go to the next slide here. There we go. Okay. So this is 3 cells now. And you can now see the spines along the cells. Okay. This is 3 neurons. Now, each neuron has like hundreds of thousands of synapses. Okay. So it's the world's greatest computer. And so those are the cells we're gonna focus in on to find out where LSD acts, because as I said, I wanted to study receptors, and we need to find out where those receptors are first in the brain. Okay. So to do this, we made mice. I was mentioning this today. I'm not going to show too much data, but I'm really excited about this. So we made mice where using CRISPR technology, which all the students should know about, we were able to tag the receptor with a fluorescent protein so we could see it. Because the receptor is present at such a low concentration that it can't be seen. And there turns out there are no antibodies that are useful for receptors. So you have to tag them. So we tag them, but we can visualize them. You can see them there. And we use this amazing microscope where I'll just tell you how the experiment is done here. This is for the students, okay? So I'm trying to convince 1 or 2 students to come to my lab. So this is the sales pitch here, okay? So what we can do is we can make these mouse brains transparent to light using a technology called optical clearing, okay? And then we use this very fancy microscope. We basically make optical sections through the brain and then do 3D reconstructions. And so this is a series, and these data sets are humongous. I mean, these are humongous data sets. But we're basically going from the bottom of the brain to the top of the brain, visualizing where the receptor is. And if we took, this is a single optical, this is not a physical, you're not physically slicing the brain, we're slicing it with a laser. So it's an optical slice, it's not being physically cut, but we take a picture of it, and we can reconstruct it. So this is a single optical slice. And just to give you a sense of the detail, it's extremely detailed. And now you can see these pyramidal neurons here lighting up, okay? This is in a little more detail here. And you can see it's in this region here of the pyramidal neuron. It's called the apical dendrite. And before I found the receptors in this area, I'd never heard of the apical dendrite. So I started reading up about it. And these are these are other pictures. So this is a Golgi stain here. This is the sort of stain that Romani Hall would use. And here's this apical dendrite here, and that's what's labeled here. So that's where the receptor is. It's in that part of the cell. For those of you who the physicians may remember some anatomy. These are layer 5 pyramidal neurons. So these are in the brain. These are extremely important neurons. And the reason for that is that these layer 5 pyramidal neurons synthesize information from all over the brain. So your view of reality is filtered through the apical dendrite of these pyramidal neurons. Okay, and what psychedelic drugs do is they cause these neurons to fire in a very it's called asynchronous fashion, but it means chaotic fashion. So what psychedelics do is they basically just induce noise in the brain. It's just noise. And for reasons that are still unclear, this noise has meaning, people, okay? But it's just noise. So it turns out it has tremendous meaning and healing potential. But it's really just static in the brain. And the brain basically just makes up a story. And that's the psychedelic experience, okay? It's just a made up story. It doesn't, so I sometimes give these talks. And some younger person will ask me about Terence McKenna. Anybody here ever heard of Terence McKenna? Any psychedelic enthusiasts? So Terence McKenna was a huge proponent of DMT, and he believed, and you can see it on the internet, that as George Bush would say, that psilocybin is a gift from the aliens. And it's a way that they communicate with us. So that's a made-up story, basically. His brain, psilocybin is just a fungus. It did not come from outer space. But this is what they look like. This is a 3D reconstruction that I did, you can see, many years ago. On a sort of very primitive confocal microscope, but I just show it because it was like, it's a really cool way to do it. Okay. So we're going to zero in on these pyramidal neurons now. So I've convinced you that that's where the receptor is. It's in these. And there are 10,000 of these synapses or spines per neuron, 70 trillion of them in the brain. And what we're going to do now is look at these. Okay. So I have this amazing PhD student, Blake, who has been able to visualize, you take my word for it, but the receptors are in the spines, actually. And that's the business part of the neuron. If you wanted to affect synaptic transmission, that would be where you would go. Okay. So they're at these spines. And if we blow up a spine, this is a cartoon. So this is very similar to the cartoon that I saw when I was an undergrad, basically. We're pretty much the same thing, except now there are more receptors, okay? So there are 15 different serotonin receptors, for instance. And so for much of my career, take a drink of water here. It's been focused on purifying these receptors and trying to visualize how psychedelic drugs interact with the receptors. Okay, because that, when I heard from this guy, that was the one thing I wanted to know. Okay. So we did it. So my first paper on this was, ancient history. But I like to show this because this was the first molecular model of LSD binding to receptor. Have any of the students ever heard of Hamilton Morris? Any of the students here? No? No? Really? Okay. So there's an amazing show you might want to watch called Hamilton's Pharmacopoeia. So Hamilton is a chemist. And in each episode, he shows the chemistry of a different psychoactive drug. And then he takes it. So this is a video. So he visited my lab. And this is the very last episode of Hamilton's Pharmacopoeia. The title of it was Ultra LSD. And the title actually comes from a paper we published. But the LSD does not stand for LSD. It stands for large scale docking. So it's a chemistry thing where we were able to identify, hundreds of thousands of new potential psychedelic drugs. So he was really thought that was really cool. So, but as part of this, he had you can see Ribosome Studio make an animation based on my work of how LSD binds to the receptor. So we're going to see it right here. And there's a soundtrack too. So for you old people like me, the song may be familiar. So that's LSD. That's what LSD looks like. So it sort of looks like that. It's a cartoon of LSD. And imagine so imagine you have taken LSD and it's floating into your brain there. And then in the background, that big white thing, that's the receptor. That's what it looks like. We saw the structure of the receptor. It binds in a multi-step fashion, which is sort of shown here in cartoon fashion. And then actually a lid comes over the top of the receptor and traps LSD there, so it doesn't get. Then the receptor is activated. And what I like to tell people is in your brain, the light does not go on. Now we're going to dive inside the cell, and somebody asked me about G proteins. Here are the G proteins. They're going to dissociate. And in the background here, you can see this spark. That's calcium. That's the psychedelic experience. That's it. Everything else is just a made-up story in your head. Okay. So I just have a few minutes left. I don't want to go over time. I want to make sure that the students particularly have a chance to ask me a question or two. I just want to. So I am not. The psychedelic research is not the thing I'm famous for. Okay. But the thing I'm famous for is like really complicated to explain. But I'll try to give you an idea. So the idea is this. There are, thousands of different types of cells in the brain. How can we ever figure out what each cell does? Okay. And so there have been many technologies that have been developed to do this. CRISPR technology, Galvan, Charpentier, synthetic antibodies and proteins, David Baker, these both got the Nobel Prize, and then opto and chemogenetics. Okay, so optogenetics was invented by Carl Weisroth. I invented chemogenetics. So this is the big thing, basically. And what it is, the idea is very simple. It was a very simple idea. Let me just go back here. So this is the idea. We think that these particular cells in the brain are responsible for LSD's action, okay? So the idea was we'll make an artificial receptor we'll put it in those cells and we'll activate it with a drug that does nothing except activate the artificial receptor. And we should be able to reproduce the effects of LSD if that's the case. Okay, so that was the idea. And as you can see, I've had this idea for a long time, but we were able to do it. We were able to succeed. I eventually got funded. And I'm just going to show you one experiment here that we did. This is sort of a dramatic experiment. So basically, this technology is used by thousands of labs around the world to understand how the brain works. And the last time I looked, there were like 23,000 papers citing or using the technology. And as I mentioned, we have sent out 59,000 orders and matches. This is like a really widely used technology. And it basically, any behavior that you can imagine, or sensation or perception, can be turned on and off using this technology. So what we're going to do here is, this is a mouse. You can see a food pellet there. And what we did was that we invented a new one of these and we were testing it out. And the idea was, we're going to put this in cells in the brain that inhibit eating. Okay? So there's this, in the mouse's brain, there's this small, there are hundreds of different sites in the brain that affect eating, but we focused on this one, they're called SIM1 neurons. There are like 100 of them in the brain. And what we're going to do is silence those neurons using this technology. So we can either turn those on or turn them on demand. And we wanted to know, if we silence the neurons that suppress feeding, what does that do to feeding? Okay, so this is a mouse that is well fed. Okay, and so we put one of those in the mouse's brain, and it speeded up. So here we get the mouse saline, and you can see. So for those of you that are not mouse scientists, the first time I presented this to my psychiatry colleagues, they thought that this was the behavior, basically. This mouse, this is a mouse grooming himself in a cage, basically. This is what mice do. But notice he's not paying any attention to the food. Okay. So he's just hanging around doing this mouse thing. And now we're going to give him the drug that activates the, this is interesting sidelight. Remember salvinorin A? The reason that it only has a 10-minute effect in humans is it's metabolized to salvinorin B, which is inactive. So we created a receptor that is activated by salvinorin B. Okay. And so we give it to the mouse. And the mouse will voraciously eat until the mouse dies or until the drug wears off. Okay. And we can do that with any behavior. Anything you can imagine that's regulated by the brain or the nervous system has already been interrogated with this technology. And I'm just going to end that. I just want to talk about the downside for example, since I'm running out of time. But one of the jokes that I make, and Matt will like this, is that, so sometimes people will ask me, why is it that we need to fund scientists? I mean, what good is science? And I say, oh, it's to keep people like me off the streets. And they say, why is that? And I say, because I have invented the technology caused the zombie apocalypse. Okay. So we have, anybody here ever seen a zombie movie? Yes? Okay. So we've identified the neurons in the brain that cause insatiable eating, murderous aggression, no sleep. And what else do zombies do? They stagger around, we can make that stagger. Okay. And we can do this with a virus. And so literally the technology is available. I could create the zombie apocalypse if I wanted to. And that's why you need to give me money. And as proof of this, I wasn't going to tell this joke, but I told it last night, so we thought it was funny. As proof of this, if anybody here has seen a zombie movie, what is the very first scene in a zombie movie? A virus is released from a government-sponsored. So I told people, if nothing else, it will be. So what's the downside? Well, they're illegal. Okay, that's like a huge problem. So don't take them. They are not approved for clinical use, as far as I know yet. Is that still true, Matt? Yes. Yes, still not approved, but they are approved in Oregon and Colorado. So I live part-time in Oregon, and you can. There are certified psychedelic therapists, and I can't recommend any. I don't know about it, but they exist. I have a good colleague, close colleague from Australia. So Australia recently legalized psychedelics. They're legal, basically. And he had severe treatment-resistant depression, and it transformed his life. He's still sort of. He's good. I discourage individuals from experimenting with psychedelics strongly. Do not do it. I've been around enough. I've seen some really bad outcomes with psychedelics, so I do not encourage anyone to take it. Except, if you're in a clinical trial and you have informed consent, that's one thing. But don't take them at a party. There are some pretty rare but very serious side effects. So because of what I study, I get anybody who has a bad side effect of psychedelics, they send me. I mean, literally, it seems. This is a bad one. This is called hallucinogen persisting perceptual disorder. This is very rare, but occurs after a single dose. So I've been contacted by younger people who have this experience of like snow in their visual field all the time, and after taking LSD one, and there's nothing you can do for it. Okay? So it's bad. It's rare, but it's a really bad side effect. The thing that I would, since I practice psychiatry and specialized in schizophrenia, this is what I would see a lot, actually. So I was trained at Stanford, and the Grateful Dead, they're a San Francisco band, and whenever they enroll it, and I moonlighted at this psychiatric ER, and we could tell when the Grateful Dead were in town because the ER would be flooded with people with psychosis. So I mean, it definitely occurs. It's not that common, but it could be really bad. And I had, in my years of practicing psychiatry, I've had not a few patients report to me that their symptoms started after they took LSD. They took LSD once, they were able to say. So again, this is rare, but it does occur. So it's not like these are innocuous substances. So they really do show promise, though. I would say particularly for depression, PTSD, obsessive compulsive disorder as well. I've heard some amazing accounts, and cluster headaches. So there's this, I don't know if anybody here has migraine headaches or cluster headaches, frequently intractable. Single dose of psilocybin, there is a lot of people with their cluster headaches. It's quite amazing. They're generally safe, but have serious, uncommon, long-lasting side effects. And this is what I do, is try to understand how they're working. And we're using this medication, or this information, to design safer and more effective medications. And our goal is actually to make a drug that is not psychedelic, but has the beneficial effects of psychedelics. So this was funded by DARPA, Defense Advanced Research Projects Agency. For those of you who are not familiar with that agency, they fund impossible projects. So they gave us an impossible project, come up with a drug which is not psychedelic, but which has the beneficial effects of psychedelics. We did. We won. And we're trying to get it into, trying to do the clinical trials. So we'll see. People ask me, is it possible? I say, so this is, now you understand the figure, right? Students, where are the students? Yes, 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 Patty? Kind of. Yeah, not so much. All right. And this is the team of talented postdocs from my lab that did a lot of this work, as well as my main collaborators. I like to highlight Dave Nichols, who's like the grandfather of psychedelic research in the US, just a wonderful guy. And then everything is funded by your tax dollars. So, and that's it. I have time for questions. I'm sorry I went over time, but so okay, thank you. I only want questions from the students first. Okay, so let's hear from the students. Any questions at all? Where's one? Way in the back. Okay.
Making a drug that's not psychoactive, but it's presumably activating this receptor. How do you keep it from making that stack that is activating it? How does it?
Yeah, that was the key. That was the trick, was finding out how to do that. So we didn't know when we started out. And it turns out that you have to just activate the receptor just a little bit, a certain pathway, basically. But what I say is, in mice, they're not psychedelic. Okay, so humans, another story, but at least in mice, they're not. So so we were able to make these compounds that just tickle the receptor just a little bit, not enough to cause a psychedelic experience, but they cause this increased spine formation, which we think is, and we give a single dose to a mouse. And we have these mice that are genetically engineered to be depressed. And they're no longer depressed for like weeks or months afterwards, just one dose. Yeah. So if you're a mouse, we can cure your depression. All right, other questions from students? Yes.
Indeed, I believe it was called chemogenetics, the technology you invented. You said that you made a receptor that activates to the B form of the drug you first talked about in the beginning.
It's activated by that, yeah.
Do you have to administer the drug in its A form to the mouse, or do you
No.
metabolize it?
No, we administer the B form.
Okay.
We administer the inactive form. Yeah. Yeah. Yes.
In your paper, you highlight the importance of the receptor, the 5-HTH2A receptor, and its affinity, with GQ or arrestin pathways. So could you kind of elaborate on what type of behavioral patterns or other therapies we can see in the difference? I mean, if the receptor depends on the affinity of what they kind of like connect to, what difference could we see on like therapies or behavior?
Yeah, that's an amazing question. Wow, that's great. That's a really insightful question. So we don't know. I wish I could give you an answer to that. So the problem is we're not able yet to make drugs that are so exquisitely sensitive for one pathway versus another. But my lab recently discovered something that I think will help us with that, and we can answer that question. That's like literally one of the questions. You want to be a researcher?
Yes.
Oh.
Yes. I have another question real quick. So you talk about a lot of the serotonin 2A receptor, because it's the one that is activated by these drugs.
Right.
So I'm kind of like very conflicted with the use of psychedelics, especially for patients that have, because of the affinity to the 2B receptor as
Right,
well.
right,
The patients with, cardiac, blood fibrosis.
yes.
So, I mean, how do you see this type of research and therapies implemented?
Yeah, so this is like a really phenomenal question. So there are all these serotonin receptors, there's the 2A, the 2B, the 2C. So psychedelic drugs activate all of those, the known psychedelic drugs activate all those receptors and even more. And the 2B, drugs that activate that cause valvular heart disease. Very serious valvular heart disease. And my lab discovered this some time ago. And so the FDA has alerted to that as well. The FDA has guidance out now on that. So the concern actually is with microdosing. So for therapeutic purposes, people are basically taking 1 or 2 doses. But microdosing, there is a risk there of serious, potentially life-threatening valvular heart disease. We just don't know enough. Nobody has done a study on it. But what we're doing is our drugs don't hit that receptor. So, not a problem. Right, yeah. So we basically invented, psychedelics hit, 30 receptors. Our drugs hit 1 receptor. That's it. Nothing else in the brain. They're highly selective for that receptor. Yeah.
Fascinating.
Yeah. It's, that's the really cool stuff we do, but it's just so intense. I just can't get into it here. Yes.
When you say that it kind of tickles the receptor, does it not bind multiple times?
The bind's just fine.
Well, yes, but you talked about with LSD that it goes through like multiple binding configurations before fully being locked in.
That turns out to be irrelevant to the psychedelic activity.
Okay.
But it does explain why LSD lasts so long. The effect. Because once LSD is on the receptor, it's on there for 8 hours, it's not coming off. Okay. That's another reason not to take LSD. Yes.
Okay, so, I think you kind of, you talk about the, studies that have been conducted with, psilocybin. I was reading about, one study conducted by NYU. It was like 3 universities with 92 participants, and they demonstrated that all these participants have cancer, and they have, end-of-life depression. So it demonstrated that after 1 single dose, 80% of the participants were in remission, like
Yes.
the second day,
Yes,
which is amazing.
it
So
is amazing, yes.
my question to you is, with your experience in the pharmaceutical, realm and everything that entitles to patent new medication and therapies, if a drug like that, like yours, that is so effective, potentially,
In mice.
yes, potentially that could be translated to human beings, and that would be, an amazing discovery. But if that drug hit the market,
Yeah.
how do you see, or what are your expectations as far as healthcare access? We have such a huge issue.
So DARPA is really concerned about that as well. And that's why so we had a company basically that was commercializing these, and we've now bought back the patents. And what DARPA wants to do is to make sure that doesn't happen, basically. They want to make sure it's affordable. So this is going to be, with all the chaos, sadly, with all the chaos going on right now, from one day to the next, I don't even know who's working at DARPA, literally. But the plan is to have this totally funded by the Defense Department, and then to make it freely available to veterans, basically, or nominal cost. And also, it'd be hopefully nominal cost to, you and I. So this is the vision, basically. So the vision we had at the beginning was, so DARPA likes to fund these impossible. They say, we fund the impossible, but projects that will change the world. That's their sweet spot. So the internet, it's called DARPANet. Stealth technology, quantum computing, artificial intelligence. They pay for a lot of that. the Nobel Prizes this year, David Baker's work. Okay. So, their idea was, can you come up with a drug which is effective like a psychedelic for depression, anxiety, post-traumatic stress disorder, substance abuse, without any of the downsides of psychedelics? Because the idea is that would transform the world, right? You know, take a drug, it does nothing to you, the next day you're fine, right? That would be transformative. So, and as part of their mission, they want to make sure that it's available to their, they call it war fighters, veterans. So, yeah. So, yeah. So we'll, you know, we're hopeful. I don't know if it's gonna work. It's research. Usually it doesn't work, but. Okay, other questions, students, any student? Yes, in the back.
What is the mechanism such that this is such a permanent or long lasting effect? How is it important? If it's reading static, is it permanently activating this receptor?
No. So we don't know, but what we think, so this is one of the things we're trying to find out. So, you see this neuron before psychedelics and after psychedelics, it has all these branching. So it increases these connections in the brain, and those are long lasting. So there are studies in mice now showing that a single dose of psilocybin basically induces permanent changes in the wiring of the brain. That's basically it. We don't know why that is. It's very unusual. I think we sort of know, but it's a very controversial area in the field right now. But this is basically it, that it changes, basically causes a physical change in the architecture of the brain, which has been, yeah, for better or worse, right? Okay. One more question, and then because we're over time here, and I can see people nodding off. Yes, the students.
Could your technique for tracking the activity of receptors in the brain also be applied to other biomolecules like mRNAs or transcription factors, or is it receptor issues?
So chemogenetics can be applied to any protein. We can turn on transcription, so we can turn it on, turn it off, but we're not yet able to say design an RNA that binds to, you know, X, Y, or Z. But literally any protein, chemogenetics is sort of a generic. Literally any protein can get at. Okay. All right, thank you very much.